Welcome to Biology at Priyojit, my dear viewers. Today we will explore restriction enzymes and DNA modifying enzymes, the essential molecular tools of genetic engineering. From different types of restriction enzymes and their recognition sequences to sticky and blunt ends, we will understand how DNA is precisely cut. We also discuss key DNA modifying enzymes like DNA ligase, DNA polymerase 1, nuclease, and end modifying enzymes that help join, synthesize, and alter DNA. Get ready to discover how these powerful enzymes drive modern biotechnology and recombinant DNA technology. Restriction enzymes, also known as restriction endonuclease, and from its name we can predict that it's an enzyme which cut the nucleic acid, and as it is a nucleus and it cut the nucleic acid from its intermittent site, that is why it is called as endonuclease. So it also known as molecular scissor. This is the image showing the activity of a molecular scissor. It cuts the DNA. It was discovered in 1962 by Smith and Nathans. Occurred naturally in bacteria, and in bacteria they function as a defense systems against bacteriophage infection. They cut foreign DNA also. There are approximately 3,500 type 2 enzymes discovered, and among them 200 are commonly used in genetic engineering laboratories. So, host control restriction and modification. Host control restriction, bacteria basically destroy invalid viral DNA and it is carried out by restriction endonucleases. Modification system, host DNA is protected by methylation and it is done by DNA methyl transferases. It prevents self-digestion. Methylation occurs at adenine 6 methyl adenine. Cytosine, in case of cytosine, it is found in 5 or 4 methyl cytosine. So the restriction enzymes and methyl transferase, both of them form the restriction modification system. Types of restriction enzymes: type 1, type 2, type 3 and type 4. Type 1 restriction enzyme, they basically recognize specific sequence but cut at random distant sites. They travel nearly 1000 to 5000 nucleotides. They cleave one strand first and then second strand. They create a gap that is approximately 75 nucleotide long. Cofactor magnesium 2 plus ATP and S-adenosyl methionine SAM is required for their enzymatic action. Structure, they have three subunits like R, M and S. R subunit is responsible for restriction cutting, M subunit is for methylation and S is responsible for substrate specificity. So basically they are not useful in genetic engineering. Here the image showing the protein structure of three different enzymes. This is type 1, which is a multi-subunit complex. Type 2, there is also multi-enzyme complex, and type 3 is another multi-enzyme complex. So these are the color showing the modification, recognition and restriction site. Type 2 restriction enzymes, they recognize specific target sequence and cut within or near the recognition site. They produce defined fragments. They only require magnesium 2 plus for their enzymatic activity, and most they are widely used in genetic cloning. Recognition sequence, there are 4 to 8 base pair long. They are usually palindromic sequence. Example 5 prime GAA TTC 3 prime and 3 prime CTT AAG 5 prime. So why it is palindromic? Enzyme act as a dimer. It has two-fold rotational symmetry. Type 3 restriction enzyme. A type 3 restriction enzyme cleave at defined site. It requires ATP, Mg2 plus and partial SAM, S-adenosyl methionine, and it is the intermediate type between type 1 and type 2. Type 4 restriction enzymes, they recognize and cut methylated or modified DNA. They act against modified bacteriophage DNA. So not part of classical restriction modification system. Examples include MCRA, MCRBC and MRR. Nomenclature of restriction enzymes based on Smith and Nathan's 1973 proposal. Naming rules: the first letter of an restriction enzyme indicates the genus, which is italic and capitalized, and the next two letters indicate the first two letters of the species, which is also written as italic. The third point is the strain designation. that is R, and the Roman numeral is the indicates the order of discovery of that restriction enzyme. So eco R1, E means Escherichia, Co means coli, R is the strain RY13, and I1 indicates the first enzyme discovered. So other example, hint 2 Haemophilus influenzae, BGL1 Bacillus globigi, etc. So what is the frequency of recognition? Recognition site recognition depends specifically on the sequence length. If the recognition site is 4 base pair long, 6 base pair long, or 8 base pair long, so respectively the expected frequency will be once in 4 to the power 4, that is 256 base pair, once in 4 to the power 6, that is 4096 base pair, and once in 4 to the power 8, that is 65,536 base pair. So shorter sequence always more frequent cutting, and longer sequence has less frequent cutting. That can be easily predictable. So nature of cut ends. There are basically two types of cut. Blunt end or flash end, which form a straight cut. There is no overhang. Example, He3, SMA1, etc. Advantages include any blunt ends can join, and they are useful when no extra sequences is desired. Sticky or cohesive ends, they are also known as staggered cut. They produce overhang. Examples include EcoR1, BAM-H1, and HINT3. Advantages include complementary pairing and more efficient ligation, and they are preferred in gene cloning experiment. DNA modifying enzyme, all enzymes that degrade, synthesize or modify DNA or RNA, they are DNA modifying enzymes other than restriction enzymes. They are essential for DNA manipulation, recombinant DNA technology, gene cloning, etc. So DNA ligase is basically act as molecular glue. It function, it joins DNA fragments, it repair phosphodiester bond, and it seals sugar phosphate backbone. T4 DNA ligase, it is most commonly used. It requires ATP. It joins sticky ends efficiently, and the blunt ends are less efficient. They works on double-stranded DNA and DNA RNA hybrids. They have no activity on single-stranded DNA. So this is the image showing the activity of the DNA modifying enzyme, that is in the DNA ligase, which acts as a molecular glue. Nucleases, this is the image showing the three-dimensional structure of nuclease. It cut within DNA. Example, restriction enzymes, S1 nuclease, etc. Exonuclease always remove nucleotides from the end sites. So there are basically, this is the image basically S1 nuclease. It is specific for ssDNA and ssRNA. They are most important nucleases in genetic engineering. It removes single-stranded overhangs. Other important nucleases used in genetic engineering are DNase1, exonuclease 3 from E. coli, lambda exonuclease, BAL31, BAL31 exonuclease, etc. DNase1 cuts both ssDNA and double-stranded DNA, which are the random cleavage and used in controlled digestion. Exonuclease 3 from E. coli has 3' to 5' exonuclease activity. They produce DNA within protruding 5' ends, and they are used in deletion studies. Lambda exonuclease, that is, it has 5' to 3' exonuclease. It removes nucleotide from the 5' end and produces 3' overhang. BAL31 exonuclease, it has fast 3' exonuclease, which slow endonuclease activity and shortens DNA from both ends, used to generate deletions. DNA polymerase 1, that is derived from E. coli, whose activity are DNA polymerase activity, that is 5' to 3', 5' to 3' exonuclease activity, and 3' to 5' exonuclease activity. Its uses are following: nick translation, DNA repair, and radio labeling. Clena fragment is basically modified DNA polymerase 1. They lacks 5' to 3' exonuclease, and they retain polymerase activity and 3' to 5' exonuclease activity. Their uses are DNA sequencing, filling sticky ends, recombinant DNA formation, etc. Reverse transcriptase is RNA-dependent DNA polymerase. It converts mRNA to cDNA, and no exonuclease activity is there, and they are essential for cDNA synthesis and eukaryotic gene cloning. So this is the image showing the Clena fragment. Enzymes modifying DNA ends are alkaline phosphatase, which removes 5' phosphate and produces 5' OH. It prevents vector self-ligation. Types, there are bacterial alkaline phosphatase and CIAP, that is calf intestinal alkaline phosphatase. T4 polynucleotide kinase, it adds phosphate to the 5' OH, that is just opposite to the alkaline phosphatase. It is used for radio labeling and preparing DNA ends for ligation. Terminal transferase add nucleotides to the 3' end and adds homopolymer tails. They are used in recombinant DNA construction. So this is the summary table. So in conclusion, we can say that restriction enzymes and DNA modifying enzymes are the core tools of genetic engineering. Restriction enzymes precisely cut DNA at specific sequences, while modifying enzymes like ligases, polymerases, and nucleases join and synthesize and refine DNA molecules. Together, they form the foundation of recombinant DNA technology, enabling gene cloning, sequencing, and modern biotechnological advancement. Keep exploring the powerful tools of molecular biology. Thank you.